All right, so in the rest of the lecture, we will cover alignment statistics. So that's how do you evaluate significance of an alignment, or when you find a homolog or a sequence that's similar to your query in the database, how do you evaluate how similar it is and whether that similarity is of significance. And then we will describe multiple alignments where instead of comparing just two sequences, you are looking at more than two sequences and trying to find commonalities between all of them. And we will describe different ways that these multiple alignments are represented as sequence motifs or profiles. Okay, so when you find two things that are similar, that similarity could be just by chance. Okay, and the chance is because they are using the same alphabet, so there will be some short matches that are similar just by chance, or it could be due to common ancestry. So it could be because of divergent evolution, where the same sequence gave rise to the two sequences that you are comparing. So the question is, when you look at two sequences, how similar do they have to be for you to claim that they are homologous, okay? Or to claim that they share the same ancestry, that their similarity is not just by chance. One way to do that is to use percent identity. When you have two sequences, usually that's the most common value that you can report. So you could say that human and whale hemoglobin genes are 87% identical. That means when you align them optimally, 87% of the positions have the same residues between the two sequences. Okay. But alignment score is actually more sensitive because now you're not only looking at the identical residues, you are also looking at compatible amino acids, right? So two amino acids do not necessarily have to be identical, but they could serve the same function in a position. Scoring matrices allow you to score that compatibility. So a percent identity is essentially you are considering a scoring matrix where the diagonals are 1 and off diagonals are 0. So anything that is identical, you are considering it compatible, and anything that's not identical, they are not compatible. So that's what the percent identity reports. Scoring matrices are more sensitive because they take into account different compatibility measures between amino acids. And that's usually for amino acids. For DNA sequences, percent identity is usually sufficient. That's why in the homework, you looked at DNA sequences when we asked you to find the percent identities, but you did alignment and found this alignment scores when you looked at the proteins. Okay. All right. To find the statistical significance of any event in general, we need a model for random similarity. Okay, so we need to build a model for things happening by chance. And then we will assess a similarity. We will assess as a given score based on how different it is from a score that is generated randomly. There are several statistical measures that you will need to know. P-value, you should all know from your statistics courses. And that is the probability that at least one sequence will produce the same score by chance. The smaller the p-value, the higher the statistical significance. Okay. E-value is the expected number of sequences that will produce at least the same score. Okay. And z-score is the number of standard deviations. So considering a Gaussian distribution, a normal distribution, you look at how different from the mean of the distribution the given score is. So that's the z-score, the number of standard deviations from the mean. Okay. So let's try to build a model for random similarities in sequence alignments. There are several models. We'll look at the most simple one first, which is the match run. And in that case, we'll only consider the identical matches. Where we look at when we incorporate substitution matrices, things will get slightly more complicated. And there's a model by Carl and Altschul which models sequence similarity scores using a substitution matrix. Okay. Whether you have gaps or not will change the random model. Okay. And the global alignments may have a different model than local alignment. So the distribution of scores will depend on whether you have a gap penalty and how much that gap penalty is, and whether you are performing a local alignment or a global alignment. Okay. All right. So let's go back to some basic statistics and ask a few questions and give you some definitions. The expected value of an event is you look at all the possibilities and multiply each of those possibilities with its probability. So this is the value of the event. That's the probability of that event happening. All right. So what's the expected value of a die roll? You roll a die. What's the expected value? What number do you expect to see when you roll a die? Any answers? Say it again. 1/6. So 1/6 is the probability of any of the numbers. It's not the expected number the number you expect to see. 2.5? 3.5. Yes. So 3.5 is the expected value that you would see from a die roll. You will never get 3.5, but that's the expected value. All right. So that's sort of if you roll the die 1 million times and looked at the average of those, it'll be 3.5. Okay. So to find the expected value, you look at all possible options. So you can get a 1 with a probability of 1/6, plus you can get a 2 with a probability of 1/6. You can get a 3 with a probability of 1/6, 4, 5, and 6. So you add all of those up. So you have 1 plus 2 plus all the way to 6 divided by 6. And that is 7/2, 3.5. Okay. So the expected value of a die roll is 3.5. Okay. So now let's look at coin tosses. Let's say you took a coin and tossed it 14 times in order. And the experiment is summarized in this figure. So black is a head. A cell that's not black is a tail. And we pretend that this is, we are not cheating. The probability of heads and tails is 0.5, 50%. What's the probability of getting 5 heads in a row? Answers? Say it again. 0.5 times 0.5 times 0.5, 5 times. So that's 1/32, right? So the probability of 5 heads, these are independent events. You just multiply the probability of each one of them to get the probability of the total event happening. So that's 1 over 32. Now, what's the expected number of times that 5 consecutive heads occur in 14 coin tosses? So you have these 14 coin tosses. How many times do you expect to see 5 heads in consecutive 5 heads? So 10 is the number of the maximum number of 5 coin tosses you can have. Well, yes, it's the maximum number of 5 consecutive coin tosses you can have. But the expected number is different. To find the expected number, you take each possibility, you multiply it with its probability, and you add them up. Okay? So you have 10 places where this event can happen, and each of them has a probability of 1 over 32. So the expected number is going to be 10 times 1 over 32, which is 10 over 32. And the reason I'm saying you have 10 places is because the 5 consecutive coin tosses could start from here. So you could have 5 here. It could start from here. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It cannot start from here because you don't have enough coin tosses left. So you have 10 locations where you could have 5 consecutive coin tosses. And at each location, the probability of having 5 consecutive heads is 1 over 32. So you start at each location, you have 1 over 32 chances of getting 5 heads in a row. You have 10 you can try that 10 times. The expected number of times that you will get 5 consecutive heads is 10 over 32. The next question is, what's the expected length of the longest run of heads in 14 tosses? So if you toss a coin 14 times, what's the length of the longest run of heads? So how many heads in a row do you expect to see? And we will answer that question in a minute. All right, probability of 5 heads in a row is 1 over 2 to the power 5. So 1 over 2 is probability of an individual coin toss. You have 5 of them in a row. So you multiply all of them, you get this number. Okay. Expected number of 5 heads in 14 tosses. You have 10 places that this can happen. You multiply it with the probability, you get 0.31. All right. Okay, so the expected number of times a length L run of heads in n tosses is the following. So you have n tosses. In the example, n was 14. Okay. The last location that it could start was n minus L plus 1. So this is the first position, second position, and this is so it'll be n minus L. So you can't if L is 5, this one let's say you have 14, 13, 12, 10, 11, 10. So the last one is going to be 9. Okay, so let me just say n minus L. All right, so you don't n is not the number that you can have the coin toss. It's n minus L plus 1. But if n is really large, okay, with respect to compared to L, then you don't really care about L so much. So if n is 1 million and L is let's say 5, you don't really care about 1 million minus 5. Okay, so 1 million is going to be more or less very similar to 1 million minus 5. Okay, so a very large number plus minus a small number doesn't really matter to you that much. Okay, so it'll be approximately the same as using the n itself. That's why you approximate it. Instead of saying n minus l plus 1, I'm just writing n here. Okay, and then the probability of a single coin toss to the power l. So that's 1 over 2 to the power 5. And then this was 10, but I will use 14 instead. Okay, so I'm going to use the n itself instead of n minus l plus 1. All right, and as n gets larger, the error in this approximation will be smaller. So 14 is relatively so 14. There's a large difference between 14 and 10, but if n were very large, 1 million, then the difference would not be as significant. Okay. All right. So that's the reason. That's the logic behind approximation. It's just to make the calculation simpler, to make the formula simpler. All right. So the next problem is what's the expected length of the longest match? So what we do is we say EL equals 1. So we write 1 in the expected number of times that this match can happen, and then solve it for R. Okay, so R will be the longest match that we expect to see, and if you take the log of both sides, you get R equals that. Okay. All right. Good. Are you guys all comfortable with the math of pulling out R? Yes. Okay. All right. So that's R. The next question is expected, well, the one that we were going to solve, but I didn't solve it there, is expected length of the longest run of heads. So we just plug in the numbers into this equation. P is 1 over 2, that's 0.5. And then N is 14. And then we find this value. So this is just going to be log 2 of 14, that's 3.8. Okay. So because of my approximation here, there's a large error. I'm introducing a large error. If you want to find the exact value, you would have to use 10 instead of 14 in this example. Okay. All right. So now let's look at two-dimensional coin tosses. All right. So what that means is we are looking at the alignment of two sequences. If you look at an alignment table, you have one sequence here, the other sequence there. So any match is sort of a toss, finding an identical ladder between the two sequences. And let's assume that we are using an amino acid alphabet with 20 letters. So that's the probability of finding an individual match is 1 over 20. So you already have an I here. The probability of the other one being an I is 1 over 20. Okay. So the probability of any of these cells having a black dot is 1 over 20. All right. Make sense? Okay. So you have 1 over 20 chances of having an identical hit. So P equals 0.05. The expected number of matches is the probability of a single match multiplied by how many times this could happen. So how many times can that happen? That is the number of times that I have a cell, and that is the length of one protein multiplied by the length of the other protein. So this protein has 10 residues, the other one has 8. There are 10 times 8 cells in this table. The probability of each cell being an identical hit is 0.05. the expected number of matches is 10 times 8 times 0.05, and that's 4. So given a protein that's 10 residues long and another protein that's 8 residues long, I would, on the average, expect to see 4 identical matches when I look at their table. Okay? So if you generate several million of these sequences randomly and count the number of hits in the table, and then take the average of those, you will find that it's very close to 4. Okay? All right, now instead of looking at an individual match, let's now find the expected number of 2 successive matches. Let's find how many times that can happen. So 2 successive matches means you have 2 identical hits along the diagonal. So if one of them is here, the other one should be here. So if this were an E, you would have HE, HE. So 2 identical dots would mean 2 successive identical residues. Okay? So we are looking for events that include these 2 diagonals. So that's, I could have one there, I could have one there, one there, one there, one there. So let's now focus on the starting positions of these 2 hits. I can't have any in this last column because if I start from here, I need another one over here, so I can't have that. And I cannot have any in the last row. So this, the number of starting positions I can have is the length of the first protein, m-1, multiplied by the length of the other one, n-1. Okay? So the smaller table, this smaller table is the number of possibilities that I can have to produce 2 successive hits. Okay? And then multiply that by the probability of 2 successive hits. That's 0.05 to the power 2. All right? So similar to the approximation that we did for the coin tosses, now let's pretend that m is really large. Okay? So when m is really large, this value is going to be very close to m itself. I mean m times n itself. So the -1 can be ignored if m is much larger than 1. Okay? And in the general case, if I'm looking at L successive matches, I need to replace that that 1 with m minus L plus 1. And again, minus L plus 1 is going to be a very small number compared to m, so I can just drop it. Okay. So the expected number of 2 successive matches is the number of locations that could happen. So that's m times n. But note that this is an approximation to the actual answer. And then p to the power L. And that gives me 0.2. All right. So if you were to generate 10 million sequences and looked at their their tables like this, you would expect to see 0.2 successive matches, all right, on the average. Yeah, push it twice. Push it once, push it again, fast. Thank you. Good. All right. Okay. So successive matches correspond to a run of matches along a diagonal, right? So that's a stretch of residues in one sequence being identical to a stretch of residues in the other one. And as I said, there are this many places that this can happen, but we'll just drop this part and just say m times n. The expected length of the longest match can be found by setting this equal to 1. Okay, so we expect to see at least one such diagonal match, and then solve for r, you get this again. Okay? All right. So now, given a substitution matrix, things get slightly more complicated because now we are no longer we can just use the probability of finding an identical hit. We need to take into account the probability that an amino acid could be replaced with another one, and also the background probabilities of having an amino acid in place. All right, so we have a constraint that the expected value in the substitution matrix should be less than 0. And the reason for this constraint is that if this were greater than 0, then if you look at an alignment, a local alignment between two homologs, that alignment would tend to extend along the edges without any biological meaning. I mean, it could extend along the edges just by chance because of the way that the substitution matrix values are. Okay. So if this number is greater than 0, then you are scoring random residue pairings with a plus score. That means you tend to extend even things that are not necessarily homologous. The expected number of alignments with a score X or higher is given by this formula, and we will see how this formula is generated. Okay, so in this formula there are two parameters, K and lambda. K is just a proportionality constant, and it takes care of the fact that the places in the table are not really independent. Okay. And lambda is, again, a factor that is related to the substitution matrix. And we have lambda because the substitution matrix does not really contain the probabilities of co-occurrences. It has a scaled version of that, so the numbers are integers. So if you look at the Blossom 62 matrix, the numbers in that matrix are integers. After you generate the co-occurrence probabilities, they multiply it with a number so that the table looks integerish. Okay, so you don't have floating point numbers in the table, just for convenience. Okay, so this is how we had derived, let's say, the Blossom matrix. Okay, lambda is a constant factor to get integer numbers in the matrix, and the sum of the observed co-occurrences needs to be 1. Okay, so if you add up all the frequencies, you need to get the value 1, and we can find the value of lambda from the substitution matrix by plugging in Qij equals this, and then adding all of them up for all 20 amino acids, setting it to 1, and solving it for lambda. Okay, so that's how lambda can be generated. If you look at the scores of local ungap alignments, so if you generate random local alignments between random sequences and keep track of their scores that you obtain, and look at how they are distributed, you will see this extreme value distribution. The extreme value distribution is very similar. It looks like a normal distribution, with the difference that for large values it trails off pretty quickly. Okay, so its general shape is like that. So for like one-sided compared to the normal distribution. One side would look like normal distribution. The other side falls off much faster than it would if you looked at a normal distribution. The peak probability is this point right here. So that's the average of the distribution. And this formula gives you the score that corresponds to the peak probability. Okay, so that's the average of the distribution, U. The probability of a score being less than this, the probability of a score being less than anything in this plot is given with this formula. Okay, so that's the formula for the extreme value distribution. You have e to the power minus e to the power minus lambda, the difference between x and this peak value. All right. And the other side of that, if you are interested in the probability that a score is greater than x, then you just take 1 minus of this. All right. Okay, so having found an alignment, let's say you align two sequences and you got a alignment score of 120. And that 120 will be, let's say, somewhere here. Then you look at 120 and find how significant that is using these formulas and what the expected number of sequences is that can give rise to that score. And that's how you evaluate the significance of an alignment. So there were some parameters in those formulas, such as the kappa and lambda. BLAST pre-calculates those parameters from the scoring matrix that it uses, and the gap penalties will change those parameters. So the gap penalties are also used, and the database size. Okay. Whereas FASTA program estimates the distribution from alignments that it gets during querying. So you run a query, you give a query sequence, it will compare it with database sequences, and it will, let's say, generate a histogram of the scores. All right. So let's say there were 200 sequences that gave a score of 90, 150 sequences that gave a score of 120, and so on. So you generate this histogram, and it fits an extreme value distribution on this histogram, and the parameters of that extreme value distribution they are used to calculate the statistics. Okay. All right. So if you have a database, that could either be a random database or the database that you are actually searching against. You can find the distribution of the scores. To generate a random database, you would have to take into account the background probabilities of the amino acid distribution. So it's what you are modeling is closer to the reality. So for instance, in your randomly generated database, you would have fewer tryptophan amino acids because they don't occur as much as the other amino acids. Okay. There are some strategies to generate randomized databases. So random database is where you generate your self-random database. Randomized database is where you take an existing biological database, let's say a genome, and you generate a randomized version of it. Okay. So the steps that you perform to generate that is you scramble it up, you shuffle sections of it. You can permute it. You can take you can permute it based on windows, or you can reverse the entire sequence or portions of the sequence. So using these operations, you can take an existing real sequence and generate a randomized database. The advantage of this is it represents a real database in a more reliable way. Okay. So this one implicitly would take care of the amino acid distributions because you're never introducing a new amino acid in it.
The randomized databases, do they take into account the Markov model probabilities, or do they have?
No, it's completely random. You just chop it up, shuffle it, reverse some parts, and keep others. Okay. So this is just sort of a way to generate something that's random, but that it still has the same features of a real biological database. Okay. All right. So this figure shows that if you change the gap penalty, the distribution of the scores will change. So not just the values, but you're also affecting the distribution of the scores that you get. So as you reduce the gap penalty, the scores will naturally shift to the right because when you have less gap penalty, that means you are penalizing gaps less. Your total score will increase, but the shape of the distribution also changes. So you see a more normal-like shape, and here you see more extreme value-like shape. Okay. All right. So we said that random databases take into account the background distributions of amino acids, and here are some statistics about amino acids. The two most abundant amino acids are leucine and serine. The rarest amino acid is tryptophan, and that's W. The reason is that it's a very large hydrophobic amino acid. It's more difficult to accommodate it in a protein structure. Okay. Followed by cysteine, which is only 1.7% of the proteins. Just looking at the residue composition of proteins can be helpful and can be informative for the functionality of the protein. For example, the metallothioneins have a 30% content of cysteine. And these form metal cages by using disulfide bonds between the sulfur groups. Okay. And some anti-freeze proteins have 50% alanine, which is responsible for hydrophobic interactions. Okay. All right. Okay. So far, we've covered pairwise alignment and alignment statistics. But now we'll move into multiple alignments and alignment profiles or sequence profiles. So why do we do multiple alignment? We use it to improve confidence in homology. If you find two things that are similar, you may not be convinced that they come from the same ancestor. But if you find sort of the same sequence appearing in 20 long sequences, then you have a higher confidence that that pattern or those sequences are coming from the same ancestor. Okay. So there's less probability that those sequences have become similar by chance. We also use it to correct an alignment. So what does that mean? The alignment that we find finds the optimal score, the highest score, but the scores are based on substitution matrices, which are it's just one way of modeling the similarity or evaluating the similarity, but it's not necessarily the most biologically meaningful way. All right. So the residue correspondences that you find between two sequences may change depending on additional information. All right. So let's say you look at the structures of those two proteins, and things that you thought were corresponding to each other are in different places if you look at the structure alignment of the two proteins. Okay. So the alignment that you find by finding the highest scoring alignment is not necessarily the most biologically meaningful one. Okay. And when you use multiple sequences, you are increasing your chances that the alignment, the correspondences between residues is the correct one. Okay. So multiple sequences give you additional information that you can use to refine the alignment. Finding conserved regions. So when you look at just two sequences, when we did the NW line, you saw those pipe symbols that showed you which parts are identical, and the colons showing you the compatible amino acids. So that gives you sort of a visual idea of which portion of the two sequences are very similar. Okay, but there were a lot of identical residues in there. When you look at multiple sequences, the chances that you will have all of those to be identical goes down. And only only the residues that are most important for the protein's function will be conserved. You will see those being identical in a multiple alignment. Okay. Multiple alignments are also used to estimate evolutionary distances. So you can calculate the amount of time, evolutionary time it took for one sequence to mutate into another one, in the context of all the other sequences that you are using in a multiple alignment. Okay. Another use is in prediction of secondary and tertiary structures or other functions of a protein. Just using single sequence statistics is not as powerful as using the profiles that you get from multiple sequence alignments. So a conserved location, if a conserved residue will give you more information about what that protein does and how it will fold. So visually, you have on the left a set of sequences, and their alignment is essentially putting gaps in the correct places. So it's about finding where those gap characters should go. So you are aligning the sequences such that the corresponding residues align with each other. And similar to pairwise alignment, you want to find an alignment that gives you the maximum alignment score. And we will discuss what that alignment score means. Okay? So it's not as obvious as it was in pairwise alignment. So you will have, when you look at multiple alignments, some residues that are identical across all sequences. Okay, so this third column is C for all sequences. The first one, there are some variations, but there's a pattern of hydrophobic amino acids. And the third one also. So this is the fifth residue. So these are conserved residues, okay? The tryptophan is identical across all proteins. And then you have some conserved regions. Those are blocks of amino acids that are identical. All right. So you could have a single residue that is identical, a single column, or you could have multiple identical columns that are clustered together, and those would be conserved regions in the protein. Okay. All right. You can summarize a conserved pattern of residues by something like this. So you say, I have a hydrophobic amino acid, and then there's no real pattern in the second column. There's no real conservation or constraint on the second column. So I just have an X there. It could be any amino acid, followed by another hydrophobic amino acid, followed by anything, another X, followed by C, all right, followed by a bunch of amino acids, followed by, this should be a W, okay. So let's make that a W so it reflects C in the alignment. Okay. All right. So if you have a multiple alignment, this is more difficult to process for us, okay, or for the biologist. We would usually talk about somebody who is a biologist, and they would need to have simpler things than a multiple alignment like this. So a pattern is a lot more useful. It's more concise representation of what's important for that protein. Okay. And we will see different ways of representing patterns. Some of them are readable, some of them are more numerical, and we don't really care so much about what those numbers are. All right, so how do we generate multiple alignment? We can actually do that by repeating the pairwise alignment of two sequences. But now, instead of aligning a sequence to sequence, we also need the ability to align a sequence to a set of sequences and a set of sequences with another set. So we need to develop a strategy of order of alignments. So what we could do is generate a phylogenetic tree like this, okay? And then pairwise align two sequences, get C, pairwise align the next two sets, and then do that, okay? So we, actually this is about evaluating the multiple alignment rather than generating it. Another way is a star schema, where you are relating each sequence to a central sequence. So there will be, let's say, one sequence in the multiple alignment that forms the center, and everything else you are comparing it with that. So if I go back, I said that we want to find an alignment that maximizes the alignment score. For pairwise alignment, you just added the scores of individual pairs of amino acids, right? But for multiple alignment, we need to compare pairs of sequences and then add their alignment scores. So in a phylogenetic tree schema, you would compare E with C, find its alignment score, store it somewhere, and then C with F, find its alignment score, store it somewhere, and then do that for all such pairs, and then your total alignment score for that multiple sequence alignment will be the sum of all those alignment scores. Okay? So this is one way to evaluate a multiple alignment score, and the algorithm that you are employing to find the best multiple alignment should give you the one that maximizes this score. Okay. All right. Another way to organize the pairs and find their alignment score is by a central schema, where you pick a sequence that's most similar to all the other ones and find the alignment scores of all of them with that central one and add them up. The more common one is all pairs, where you calculate the alignment score of all pairs of sequences, and your total score is going to be the alignment, the scores of all of those pairs. Okay, so these are different ways to evaluate a given multiple alignment. How do we actually find the multiple alignment that will give us the optimal result? So let's say we have 3 sequences instead of 2. When we had 2, we built a table and a 2-dimensional table, and we filled in the scores, and we went back and followed the path to get the alignment. When you have 3 sequences, the third one is just another dimension. So now you need to build a 3-dimensional cube, a 3-dimensional table that will give you all the options of a path. Okay. And for each new cell, for each new small cube, the score will depend on 7 options that you have, right? I think it's 7. So you could either come from these edges, okay? You could come through these face diagonals, or you could come from the cube diagonal. So very similar to the 2-dimensional table, but now you have more ways to come from. More ways that you could come from to the present cell. You calculate all of the options, just like the 2-dimensional case. So coming from there, plus aligning all those residues. So this is the cube diagonal, where you are aligning these 3 residues. Okay. Coming from here, so this is a gap, a single gap. You are aligning 2 residues out of the 3 sequences, but you have a gap for the third sequence, or for the first sequence in this case. All right, and so on. So in this one, we have 2 gaps, but 1 residue there. So if your alignment is, let's say, ACGT, A, gap, gap A, A, C, gap, gap. So this column corresponds to this one, right? So all three are aligned. This column corresponds to this one. You have a residue in the first sequence, a residue in the third sequence, but a gap in the second one. This one corresponds to this one right here. Okay, so you have a residue but two gaps. This one again corresponds to that. Sorry, that one is the last one was this one right here, where the first two sequences had a residue, the third one had a gap. Okay. All right. So it's the idea is similar. The idea is the same. You build a table of scores, and for each cell you use the solution from the previous steps. But now there are more previous steps because we are working in 3 dimensions. All right. So what's the computational complexity of this method, of this algorithm? Let's say each sequence has n residues. It's n cubed, right? So this, let's call k. If you have k sequences that each of them having a length of n, the computational complexity is all n to the power k. So the complexity, the amount of time and memory, if you are trying to generate the path also, increases exponentially by the number of sequences you have. So as you have more and more sequences to align, this becomes infeasible. All right, so you won't have enough time. Well, you won't want to wait that long to generate the optimal alignment. Okay. And whenever, excuse me, whenever we have a method that can find the best answer optimally, but it takes too long, and we don't have any other method that will give us the answer any faster, we use heuristics that work fast, but do not guarantee the best answer. It's a good enough answer. Okay. So we have methods that will give us good enough answers, but faster. Okay, for 3 sequences, that was this. For k sequences, this is going to be n to the power k. To be more exact, it's this one, but let's just focus on n to the power k. So it's exponential on the number of sequences you have. So one heuristic is, instead of looking at all sequences at once and trying to find the best path, we take 2 sequences at a time. All right, and we align pairs of sequences. So it's just pairwise alignment repeated. Okay. All right. So at each step, we align a sequence to another sequence, or we align a sequence to a multiple alignment, or we align a multiple alignment to another multiple alignment. Okay. So let's say we have all these sequences. All right. First, we compare all pairs. We compare everything with everything else to find how similar they are to each other. And we build this phylogenetic tree. So this phylogenetic tree contains, it tells us if we built a hierarchical tree, how these sequences would be related. And the idea is to organize the sequences into their evolutionary history. So we are hoping that this tree represents the sequences of speciation and differentiation events in the evolution. All right? And then we start from the bottom and go up again. We take the 2 most similar sequences, or any branch of this tree, and we align them. So we align those 2. That's the first alignment we perform. Okay? And then we take this group of sequences and align another branch to it. So we generate that part. We get the number 3. So number 3 is this one right here. So it's the red blue, yellow. So now we have a group of 3 sequences that are aligned. Then we generate the alignment of these two. It gives us the group number 2. And then we align group number 3 with group number 4 to get this. Sorry, group number 3 with group number 2, and that gives us group number 4, or the alignment number 4. Okay? All right. So once we align two sequences or align 2 groups, that alignment is sort of set in stone. We don't change it anymore. So we take it and then we add more things to it. All right, so the alignment of any pair, either they could be single sequence or groups of sequences, is done by the dynamic programming alignment that we covered in pairwise sequence alignment. So it's OMN, let's say ON squared. Okay? If you have n sequences, what would the overall algorithm be? Any guesses? So each pairwise alignment takes ON squared time. Now you need to figure out how many times you repeat that process. So you repeat that process about n times, right? So it's going to be ON cubed. So actually this part is ON cubed. The first part where you are trying to perform all pairs of alignments to generate the tree, that's n squared comparison. So you are comparing each sequence with another sequence. So you are comparing all pairs comparison, so that's n squared. And within each comparison, you are building the table, which is n squared. So that's ON to the 4. And if you look at the total complexity, you have ON to the 4 on one side, and then you spend an additional ON cubed time. So what would the total complexity be? Is it ON to the 7? No. So it's going to be ON to the 4, okay? Because we always consider the case where n gets larger and larger. And when n is really large, n to the 4 is going to dominate, and you won't really care about n to the n cubed. Okay, so you take the largest power, that's your complexity. All right. When we do such pairwise alignments to build a multiple alignment, the order in which we perform the alignments matters, and it will change the result that we get. So let's have these 3 sequences, DGD, GG, and DGG. Let's align DGD and GG first. So we have 2 options to do that. We could either place GG here, or we could place GG here. Okay. And then we add the 3rd sequence, DGG. So this alignment is already set in stone. We are not going to change it. The best alignment is going to be putting DGG right there. So we get this alignment, and then we add this. We get that alignment. And these 2 alignments are different. Okay. All right. But if we align DGD with DGG first, so that is set in stone, then we don't have this problem. GG will go in here, okay, because it has a higher score. All right. So if we align these 2 first, we get a different alignment than if we align these 2 first. All right. So the order of alignments matter, and if you make a mistake early on because things are set in stone, that error propagates to the end. It will mess up the way that you add the new additional sequences to your alignment. Okay. So one disadvantage of the so-called progressive multiple alignments is that the errors propagate. Okay. So a mistake that is done early on propagates to the end, and it could mess up the entire alignment. So the selection of the sequences, the ordering of the sequences is important for progressive alignment. There's a method called genetic algorithm, and in this, the problem is modeled as a population and replication problem. So it's a population evolution problem. So we are using ideas from evolution to solve the sequence alignment problem. So how do we do that? We consider each candidate solution. So we consider each multiple alignment solution, and we could have multiple of them. We have an exponential number of them, so we don't really want to consider all such candidates. We'll generate a small set, let's say 100 of them. We will perform selection from them. We will calculate their alignment scores, total alignment scores, which is fast enough to do. We will rank them, find which ones are most promising, and then we will select from them based on how promising they are, based on how high scoring they are. So we'll only keep the ones that are high scoring. Let's say we keep the top 50 most high scoring ones out of the 100. So that's the selection phase. All right. And then we perform mutation and crossover operations on those promising candidates. So mutation means we shift things around randomly in a candidate solution. A crossover means we take two candidate solutions and we use parts of them to generate a new solution. Okay, so that gives us a new population. So each row here that is represented in binary is sort of it's representing a solution to multiple alignment. All right. So after we get the new population, we perform selection again, and we repeat this process, let's say 1,000 times, or until we are happy with the alignment score. Okay. So to apply genetic algorithm, we need to be able to perform selection, mutation, and crossover events or operations. So selection is easy. We would just calculate the alignment scores for each multiple alignment. We can calculate their alignment scores, so that will give us the fitness value for that individual in the population. So an individual is a multiple sequence alignment in this problem. And then mutation is easy too. We can take any gap symbol and move it around. So that could be a mutation operation. Crossover is slightly more tricky. What we'll do is we'll look at one individual. We will cut through a portion, and we will perform the same cut in another individual. So the same cut meaning this one is cut at the third residue, third residue, third residue, third residue. So we will find where the third residue is. We'll cut it through that. In the second sequence, the third residue is there. In the third one, it's right here. Okay. And then we would take this part and combine it with this part. Okay. So we do it like that. They will have two individuals. So this portion is combined with that portion right there to give this individual. All right. So you take two solutions to two possible solutions to a problem, and you, in a way, merge them, hoping that their merging is going to give us hopefully a better solution. Okay. All right. And then you will have a number of these where you apply selection again. You will select only the ones that are most promising. And when you do that, your population, the set of solutions that you have, will keep improving and improving. They will contain more successful individuals whose scores are higher. So as a population, your scores are going to improve. And when you are satisfied, you can just pick the highest scoring individual out of that population. That will be the final alignment, the multiple alignment. All right. So in the next week's homework, what we will do is we're gonna work on a genetic algorithm, and you guys are gonna implement it in class. So each of you will be. I'll make up teams, and you will implement a function, and we'll put it together and see how it works. So let me go back to the homework problem from, the quiz from today, and solve those problems so you guys can score them. I don't think I've covered all of it, but let me just give you the answers. So in the context of BLAST, an e-value is the expected number of sequences that you would get by chance having the same score. We haven't covered the entropy, but that is the summation of probability of ith event multiplied by the logarithm 2 of pi. Okay. So if the formula looks anything like this, where you multiply p with log p, it should be acceptable. And the log base 2 is not necessary. You could have any base. HMM stands for hidden Markov models, and PSSM stands for position specific scoring matrix. All right, so you can score the quizzes, and there are 4 questions, each of them having 2.5 points. So you can give the total score of 10. And as you are doing that, let me open the homework for 2 weeks from now. It's listed as homework 4, but we will work on this genetic algorithm next week. And there's also the other problems that you should be able to solve without much help from me. Depending on how far I get now, I might change the deadline. But we will work on this exercise in class next time. So in this problem, what we're going to do is we're going to try to discover a sequence from the alignment score. So we are given a target protein sequence and its global alignment score to another unknown sequence. And we are trying to find what that unknown sequence is. Okay? And there may not be an exact solution. There may not be a unique such sequence that gives the alignment score that we are looking for. Okay. So what we are going to do is we're going to represent this problem and solve it using genetic algorithm. So we need a way to evaluate an individual. And in this case, an individual is a possible solution. A possible solution is just a single sequence, a target sequence that we are trying to find. Okay. The fitness is just going to be the alignment score of that with the given. I'm calling what do I call it? The target protein sequence. Okay. So we are trying to discover a sequence based on its alignment score. So we will pretend that we don't know the sequence. Okay. We are trying to find it. Right. So that's the problem. But we are able to calculate its fitness value. We know how good a solution a sequence is, how close we are. All right. And that's going to be by the global alignment score. All right. And if S and T are exactly the same, which is when we find the answer, the alignment score will be infinite. And if S is a cell array of sequences, then score will be a vector of their scores when you compare them with the target sequence. So there will be a hidden target sequence, and the fitness value, fitness function will return the score compared to that target sequence. Okay. All right. So the fitness function is going to be done by. Let me see how many functions I have. 1, 2, 3, 4, 5 functions. The fifth one is most difficult. So I'll have 3 of you working on the fitness function. All right, and I'll wrap around as I move. Okay, so you can read it, but next week you can work together. And if you separate into different places, you'll need to find other people who work on the fitness function. So in the next function, we will write a population initialization function, and that will create an initial population that's a cell array of strings. So a cell array of random sequences, random amino acid sequences. Okay. And we are given these parameters. The maximum length will be given, and the number of such sequences is given. So you need to generate n random sequences whose length is between 1 and max length. Right. And if n and max n are not provided, you will use these defaults. So the next three of you could work on population init. Okay. And the next function is mutate, which takes a sequence S, so that'll be a protein sequence. There are multiple types of mutations. One of them is substitute residue in that location to a random amino acid. So you just pick a location and randomly change it to another amino acid. All right. So you randomly change a letter to something else. You could delete a residue, so your sequence will be one short. Or you can insert a new residue in any location so that your sequence is one longer. All right. And you return the sequence that you just created, that you modified. Okay. All right. So the three of you could work on the mutate. All right. The population solve puts all these together, and what it does is it takes these parameters and it will pass these to the other functions, and there are some defaults to it. So you are asked to normalize the best ratio so they add up to 1. So all of these will be, let's say, 3, 5, 7. You will add them up. You will divide them by their sum to get so that they add up to 1. You will call the population init function to generate an initial population of size n. Okay, so you have n random sequences, and then you will calculate the fitness of each such sequence, calling the fitness function. And the fitness group is going to have a hidden. They will have a sequence that's hidden from everyone else, so other people won't know about it. You calculate their fitness value, so you will get a score for all of them. These are optional, so let's skip those now. You will initialize the next generation's populations to be empty. So you have the previous generation and the next generation. You will pick the top few of them as the fittest, and these will be added to the next generation. Okay, and you will also pick mutate ratio times n of them, n of the original sequences, and do mutations on those and add it to your next generation. Okay, so some individuals go as they are. Some individuals get mutated and moved over to the next generation, and some of them are the cross ratio times n of them are gonna move to the next generation by crossing over 2 using crossover. I didn't cover crossover, did I? Oh, here you go, crossover. So in crossover, you take 2 sequences and you split them randomly at 2 locations and you combine them. Right, so you get 2 new sequences from crossover of 2 old sequences. So the solve function, the population solve, initializes from the previous generation, generates a new generation, each generation having n individuals, and repeating this multiple times. And at the end, it will return the best sequence, which has the best score, and its score. Okay. And how long it took for the algorithm to, how many iterations it performed. All right. So the population solve can be done by 3 of you all the way there. All right. I'm just pointing. I'm sorry. And then we'll start over again. Kendall, you 3 could work on the crossover because I didn't assign it to anybody else. And then we'll loop over, go back to all the way to April. No, you 3. So you'll do the population solve, all right? And you two could work on the population init, and all the way in the back three, you could work on the crossover function. Okay, so everybody knows which function they'll work on, and you can get ready for it for next week. You could start writing it, and we'll complete it next week. And you will compare your notes and write a single function. Any questions? Okay. And everybody, I'll provide a central place where you will upload, and we will see how it works when we run everything else. What I will do is I will provide key files. All right, since you will be working on a single function, you will want to know how that single function works with everything else. I will provide key files, which are MATLAB executable files, so you can execute them just like any other function, but you won't see their source code. Okay, so I will provide you these functions that are already written as P files. So when you are writing your own function, you can see how it runs with all the other functions that are already written. Okay. All right. Great. We'll see you next week.